In the last lecture and also in class, we talked about cell size. And you hopefully figured out, or have definitely figured out by now, that cells need to remain small. The reason being is they have to be able to get enough nutrients in and enough waste out, and also not to freak out their DNA. They need to maintain a much smaller cell. They need a larger volume to surface area ratio so that they can maintain a healthy, happy life. Cells, however, never stop growing. So at some point, all cells need to make a decision. Do I keep growing and eventually kill myself, or do I go through cell division and give rise to two new cells from my own existing cell? So for this lecture, what I will continue on is the more practical and more probable way that cells move on in their life, and that is going to be cell division. If you remember all the way back to our very first unit, we talked about two different types of organisms. One of them was prokaryotic, one of them was eukaryotic. I'm going to start by talking about what a prokaryotic organism will do when it divides. When a prokaryotic organism, and I'm talking mostly about bacteria, when a bacteria divides, it does it through binary fission. Fission, if you concentrate on this, it means to split. So I'm splitting my cell. Binary means into two kind of pieces. So I split my cell into two pieces. I'm not going to go into a lot of depth with this. However, I do need you to know the two basic steps of binary fission. First thing, in order to be able to divide, in order for two new identical cells to come out of this, then my cell must replicate its DNA and organelles. So it makes a copy of everything. You have two copies of everything when a prokaryotic cell decides that, hey, I need to go through binary fission. The second thing that happens is that the cell will divide. When the cell divides, the end result is two new identical daughter cells. One cell becomes two cells, and they will be identical, just like identical twins. In the diagram that is on page 5 of your unit packet, I want you to look at it and look how it actually works. It's a very simple diagram to look at. In this diagram, and I'm showing it to you on the screen, you start out with a prokaryotic cell, a bacterial cell. Now, if you remember, prokaryotic cells have circular DNA. So in this, I'm only going to show that the DNA is replicating, but keep in the back of your mind that I am also going to need organelles, and so organelles will replicate as well. My circular DNA is replicated. I make another copy of it. We will talk about DNA replication right after Christmas. That'll be your first unit back. Then what will happen once my DNA has been replicated, the sides in the middle of my cell will start to pinch together. And eventually those two sides will meet and they will form two new identical cells. In the following animation, it's going to walk you through this a little bit more, so just watch along with this and take notes about the things that you see, the things that you think are important to remember on your diagram on page 5. On the flip side of this is what a eukaryotic cell will go through. Eukaryotic cells do something a little bit different, and we're going to spend most of next class talking about what they do in a lot of depth, but I'm going to go ahead and introduce it to you. Eukaryotic cells are going to follow kind of the same process. They are going to follow two essential steps, but then it gets way more complicated in the second step. So my first step, just like in binary fission, I've got to replicate the DNA and my organelles. I got to make copies of everybody. My second step is where I'm going to start getting tricky. The end result of what a eukaryotic cell goes through is the exact same thing as binary fission. So I get the formation of two new identical daughter cells. Right now, it doesn't look any different. I've replicated my DNA and I split. It's a lot more complicated in a eukaryotic organism. In step 2, you also have, so if I continue with step 2, you have an A and a B. Step 2 is broken down into 2 major steps. Step A is called mitosis. Mitosis is the division of the cell nucleus. Step B, under the whole I'm going to make 2 new identical cells, is called cytokinesis. This is the division of the cytoplasm. Mitosis is what we will spend the great bulk of next class talking about. But that's the basics behind it. It doesn't look any different from binary fusion until you start getting into the nitty-gritty. And it is an entirely different process because you're talking about an entirely different kind of organism. One little clarification that I want to put in with this. All cells go through mitosis except sex cells. Sex cells, if you haven't kind of already figured this out, is going to be a female's egg cells or a male's sperm cells. Those go through a separate process that we will talk about in our next unit. All the other cells that make up the human body are given the term somatic cells. And this is your skin cells, your heart cells, your muscle cells, your nerve cells. Any cell except for these 2 go through mitosis, and we call them somatic cells. Now I'm going to leave the steps of mitosis for class so that you're with me. You can ask me any questions you have. In order to understand mitosis, though, you got to take into account certain things. And it's largely all centered around something called a chromosome. A chromosome is your DNA. So it is genetic material that is passed from one generation to the next. At the very beginning of the year, I gave you 2 different names for DNA. When something is going through mitosis, my DNA becomes chromosomes. But if my cell is at rest, if it is living its everyday life, I call it chromatin. So this, if you remember, is DNA at rest. Your cell is just living its everyday life. And this is when you are in your nucleus. There's your nucleus, here's your nucleolus, and your DNA looks like jumbled spaghetti all around the cell. You couldn't pick anything out from it. It just looks like a jumble of spaghetti. However, when the cell starts to go towards division, my cell has gotten big enough that it's gotten that signal that, hey, you need to start dividing. You need to take care of this or you're going to die. Then my chromatin will start to shrink. And chromatin goes into becoming chromosomes. This is your DNA at cell division. This is when your DNA condenses into visible, compact structures. If I had my nucleus over here, here's my nucleolus, then my DNA starts to take on kind of an X appearance, and they will compact down so that you can see your different chromosomes. They will condense and they will compact down so that you now have some very visible structures in your nucleus. Chromosomes are usually represented by X's, and there is a very good reason of why they are shown as X's. At the base of a chromosome, they are made of DNA, wrapped around proteins. These proteins are given a specific name of a histone. So in the following picture, what you're looking at is you've got your strand of DNA, which is going to be a double helix, and we'll get into that after Christmas. It is wrapped around a histone protein, and that allows it to condense down and eventually take on a very simple X shape. And that X shape will come in very handy. And it looks a lot like this picture that I'm showing you, where you can see that it's all wrapped up, it's all very tightly packed, and now my DNA has been pulled all in so that I can get it replicated, and then I can get it separated so that I can go through cell division. So it takes on a very unique shape, and that unique shape lends to cell division. It helps allow for a very efficient cell division. Within a chromosome structure, when a chromosome is fully condensed, you can now break it down into individual parts. So on your sheet, I want you to pick 2 different colors, and I want you to draw me an X, but I need it to be in 2 different colors because there is a very distinct reason of why. This whole thing is called a chromosome. However, I can talk about it in a couple of different ways, depending on what I'm talking about. On this side, my yellow part of my X can be called a chromatid. Same thing can be said for the X side, the red X side. Both of these are chromatids. Together, I can talk about them together, sister chromatids. All this is you're talking about the fact that sister chromatids are identical DNA strands. And it means that my DNA has been replicated. When you start to condense down into chromosomes, you are finishing up the replication process of cell division. And I will have my identical strands, the 2 that I've created, they will stay together until I rip them apart. They stay together by a little notch right where they would come together, and this little notch is called your centromere. So it's just a little button, if you will, that holds the 2 chromatids together until I get to the point in mitosis where they get ripped apart and they get sent off to their individual cells. With chromosomes, one unique thing to keep in mind is that every single species has its own unique set of chromosomes. Every organism on Earth has a specific number of chromosomes. For example, fruit flies, which are the favorite organisms of a lot of geneticists, only have 8 chromosomes. We found out a lot of information by doing experiments on fruit flies. Another example, humans. We'll talk about ourselves. Humans have 46 chromosomes. Our distant cousins, the apes, have 48 chromosomes. Every organism has a unique number. And then within that, on those chromosomes, you also have unique genes that make them who they are. The way that chromosomes can be studied, and you will get to work with these in this unit, are things called karyotypes. These are a map of an organism's chromosomes. And I will conclude this lecture by showing you a couple of different examples, and you will get to work with these in a little bit. Take a look at these last two things, complete the other assignment that is part of your homework, and I will see you in class.